find because it suggests that there are many, many genes that are contributing. You see the same thing if you look at sperm head abnormalities. So normal sperm have this very pointy shape. Abnormal sperm are sort of rounded, and they have this frown because they participate in fertilizations. And what you see is the proportion of abnormal sperm goes up almost linearly with the amount of the X chromosome that's intercrossed. So we got to this point, and this was about 5 years ago or 6 years ago, and I thought, well, okay, what we found is the X chromosome is really important, but the whole X chromosome seems to be important, as if there's something distributed along the X that is somehow causing sterility. And I remember this important paper that was published in PNAS in 1972 by Lifshitz and Lindsley, suggesting that the X chromosome in males is inactivated in meiosis, in male meiosis, and that a disruption of that process might be a general explanation for sterility across lots of different animals. They were actually thinking about both Drosophila and mice when they wrote this paper. So this is important. Everybody knows about X inactivation in female mammals. Females have 2 X's, males have 1. And we know that in the somatic cells of females, 1 X is shut down. But in males, in all male mammals, we now know that the X is also inactivated, but it's just inactivated during spermatogenesis and right at pachytene of meiosis. So as cells enter into pachytene, so in prophase 1 of meiosis, the X chromosome is transcriptionally shut down. All genes on it are silenced. And you might think, well, you know, why would that be? That's kind of a weird thing. And there's evidence that a similar process happens in Drosophila, or at least that the X is a very repressive regulatory environment in Drosophila male meiosis. And so you might wonder, why would that be? And one possibility is that this has evolved as a general defense against invasion of meiotic drive elements. So meiotic drivers can invade on sex chromosomes much more easily than on autosomes, and if you shut down transcription on sex chromosomes, that may provide a general defense against drive. So this is a diagram of meiosis. Pachytene is when everything shuts down. This has been very well characterized in mammals. So I thought, well, maybe what we're seeing is actually support for what Lifshitz and Lindsley proposed many, many years, 40 years ago. And that suggests a very simple test, and that's that if we just look genome-wide in sterile and fertile hybrids, and we have this nice control, in a single cross, in one direction we have sterile hybrids, in the other direction we have fertile hybrids. So we can compare expression of those 2 kinds of hybrids and ask, in the sterile hybrids, do we see misexpression on the X chromosome, and do we see overexpression? So things that are normally shut down, are they expressed? So every once in a while you do an experiment where you just get a crystal clear result. And so for the graduate students, this doesn't happen often, but it's really fun when it does. And so Jeff Good, who's a former student in the lab, did this work. And what we're looking at are the genes that are misexpressed in the sterile hybrids relative to the fertile hybrids. Genes that are underexpressed are in blue, overexpressed are in red. And the X chromosome is here. And so 2 things should jump out at you. First, most of the things that are misexpressed are on the X. The second is that everything that's misexpressed on the X is overexpressed. And then on the autosomes, you see a smattering of things that are overexpressed and underexpressed in roughly equal proportion. So we thought this was very good evidence for the idea that sterility is associated with the disruption of normal X chromosome inactivation in male meiosis. And we followed up on this in a number of ways, which I'm not going to talk about, but I think that's true. So this tells us that disruptions of gene regulation on the X chromosome are contributing to reproductive isolation in mice, in hybrid mice. What it doesn't tell us about as much is what's going on the autosomes. And this is a very stringent threshold. There are actually many genes that are expressed differently, and if you use a less stringent threshold, you'd see many genes lighting up on the autosomes. I want to talk about what we think is going on in the autosomes, and this is work that was done by Qtl Mapping. Where's Katya? Wave your hand, so you can correct me if anything I say is wrong. And if you have any questions about this, ask Katya. Okay, so gene regulation is based on interactions. So genes may be expressed at some level as indicated by that arrow. There may be a cis-regulatory element which which binds a transcription factor, and when that happens, that may turn up the expression of that gene. So the binding of a transcription factor will turn up expression. If we now look at a different species, we might see that there's a different cis-regulatory element, and so the low level of expression that exists in the absence of the binding of that transcription factor is what we see. So mice are diploid. So now imagine we're looking at musculus and domesticus, and what we observe is simply this difference in expression level between musculus and domesticus. So in this case, musculus is expressed at a higher level. In this case, domesticus is expressed at a lower level. And the question Katya wanted to ask was, what's causing that divergence in transcription level? Is it a change in the transcription factor, or is it a change in the cis-regulatory element, as diagrammed here, or is it some combination of both? And so she did an experiment, which I like to call the Witkop experiment, because it was first published by Patricia Witkop at the University of Michigan over 10 years ago. And the experiment it's a really clever one, and this is how it works. If you take two lineages like this, where you see a difference in expression, and you want to know whether that difference is due to a change in cis or trans, you make the hybrid, and you look at the expression of the two alleles in the hybrid. And if you do that, and you see the following, so in this case, the two alleles in the hybrid look just like the differences between the two parents, then you know it must be due to a difference in a cis-regulatory element, because that's what's controlling each allele in the hybrid. They're in the same transacting environment. This transcription factor is accessible to this regulatory element, but the sequence difference prevents binding. So if that's what you observe, you would conclude that the difference is due to a change in cis. Alternatively, it may be that you see a difference between species and they have the same cis-regulatory element, and this one is expressed at a higher level simply because it has this transcription factor and that one doesn't. You now put them together in F1, you would see that this transcription factor, which is provided by that half of the genome, can bind here, and both alleles would be expressed at the same level. So that's the logic of the experiment. You compare differences between alleles in an F1 to differences between the parents. And if the difference in alleles in the F1 recapitulates the difference in the parents, you know that it's due to a change in cis. And you can imagine combinations of those or intermediate situations. So comparisons between the parents and the alleles in the hybrid helps identify changes in cis, trans, or both. And so Katya did this experiment crossing musculus and domesticus, and this is what she found. So what we're looking at here are the differences between the parents. So this is on a log scale, so this is where the two parents are equal. In this case, one parent is higher than the other. In this case, that same parent is lower than the other. And then this is the difference between the two alleles in the hybrid. So I won't walk through all the colors, but the easiest one to see is this one-to-one line, this red line. And that's the situation I described, where the differences between alleles in the hybrid completely recapitulate the difference between the parents, and you conclude that the differences are all due to changes in cis. Another one that's easy to see is this brown line, the horizontal line, where you see differences between the parents, but no differences between the alleles in the hybrid, suggesting that the differences are entirely due to trans. Now, in some ways, the most interesting aspect of this graph is this vertical line, where you don't see any differences between the parents. So this is a gene that's expressed exactly the same in musculus and in domesticus. And yet you look at the two alleles in the hybrid and you see that they're different. So if all you did was look at the parents, you would think there had been no evolution. The gene is expressed at the same level in both parents. You now look at the hybrid and what you see is, no, the two alleles are expressed at a different level. And so that must mean that there's been a change in cis that's compensated for by a change in trans. And they're going in opposite directions. And in fact, if you look at this statistically, Katya found an excess of those kinds of changes that are going in opposite direction. Now, go back to the model the Dubzhansky-Muller model I showed you at the very beginning, where we had interactions between alleles, it's easy to imagine that if gene regulation is underlying reproductive isolation in general, that those sorts of compensatory changes are exactly those that are going to be, genes that are going to be misexpressed only in the sterile hybrids. And so we looked for that in the following way. We just took all the genes and looked in the hybrids and asked if they're misexpressed, meaning expressed at a level above or below the level of both parents. And there were a very small number that were misexpressed just in the sterile hybrids, 35, and many thousands that were not misexpressed in the sterile hybrids. But now if you ask among those 35, how many of them show cis and trans changes in opposite directions as opposed to any other kind of combination of changes, and the vast majority are of this sort. So that's exactly what you would expect if compensatory evolution is leading to problems in hybrids when you bring the two things back together. So I think that these are very good candidates for hybrid male sterility. And a subtlety that you might not catch immediately from this is these genes themselves are good candidates for hybrid male sterility because the cis change is actually at that gene. And so we now have 29 genes that we think are worth pursuing and thinking about from the perspective of reproductive isolation. Okay, so that was a lot of, the next part of the talk is a little more organismal. So if you were kind of zoning out on all the cis and trans stuff, here's the summary. Now's a good time to wake up. So house mice subspecies are isolated by hybrid male sterility. It's asymmetric and polymorphic, and that's consistent with very recent divergence. The hybrid zone studies, I didn't really talk about genome scans, but they reveal a large role for the X chromosome, and lab crosses show that the genetic basis is very complex, and X-linked and autosomal regions both contribute to isolation. There seems to be a disruption of meiotic sex chromosome inactivation as proposed 40 years ago by Lifshitz and Lindsley, but more generally, compensatory regulatory evolution seems to contribute to reproductive isolation. So when I started this project, my dream was to find the gene causing hybrid male sterility, and how naive I was. There is not a gene, there are many genes, and we think a lot of the important changes are regulatory. So that's the speciation stuff. Okay, the adaptation stuff is the following. So Domesticus is the subspecies that's native to Western Europe, and starting with Christopher Columbus, it was spread around the world. So there's Mickey Mouse driving a ship across the ocean. In fact, the history of mice around the world sort of parallels the history of human migrations. And people like Jeremy Searle at Cornell have been very interested in trying to document this in a detailed way. But what this means is it actually provides a nice arena for us studying adaptation on standing variation over very short evolutionary timescales. So these mice have just been in the Americas over the last few hundred generations, and they are everywhere. I have seen house mice in the basement of VLSB that clearly came from outside. They're wild ones. They didn't come from us. So this is a big project in the lab that's involved lots of people, and we did a transect from Tierra del Fuego to Canada, collecting mice roughly every 6 degrees of latitude. So Felipe Martins, who many of you know, a former postdoc, did South America. Taichi helped. There are 2 transects that are actually not shown here, one in Bolivia and one in Ecuador. Taichi helped with one of those, with the Bolivian transect. Megan did this transect on the east coast of North America. And then Taichi did this transect on the western part of North America. The red dots show locations where live animals were collected. So Felipe collected these from Brazil. Dana Lynn collected these from Florida. Mike Sheehan collected those from New York. And Megan collected these. And Taichi collected those from Canada. So it's really been a lab effort. And I'll try to identify the people who did some of this work as we go along. But in addition to sampling 26 populations in the wild, we collected these live animals and we've used them to start new inbred lines of mice. And this is just a little bit of an aside, but all laboratory mice that are used over the entire world by biomedical researchers derived from a very limited set of founders, and representing a very small amount of the genetic variation that exists in the wild. So part of the motive for this project was to establish a large new collection of inbred lines that can be made available to biomedical researchers everywhere. And so when we are done with all the inbreeding, we're going to rederive these and give them to the Jackson Laboratory for distribution. This is modeled a little bit after some of the things that Trudy McKay has done in Drosophila, although there's some important differences as well. So we have many lines going, and some of them are as far as 12, 14 generation of inbreeding, so we're pretty far along. We took many tissues from mice in the wild, prepared specimens of all of these, and I think I've said everything else that's on here. This was not my idea. So the first person who did this kind of work was Carol Lynch. This was published in AmNat in 1992, and she sampled mice from 5 populations along the east coast of North America, and showed that there was clinal variation in body size and nest size, and that these differences persist in the lab, suggesting that there are phenotypic differences that are clinal, that are probably adaptive. When I read her paper, I was very impressed, but I didn't believe it. It just didn't seem, I didn't have a good reason for not believing it, but I thought that nest building behavior would evolve so quickly and be different between these populations. Surprised me. So we replicated this. This is just body size in Brazil, Florida, and New York, both in the wild and in the lab. And there are enormous differences. So there's clinal variation in body size. You see it in wild mice. You also see it if you bring mice into the lab. So these are clearly genetically based differences. Mice in New York are 50% bigger than mice in Brazil. And this is something that's evolved in a few hundred generations. So it's a very, very strong effect. I'm not really going to talk about this. This is Taichi's work. This is a lab project, and different people are interested in different phenotypes. Taichi has been looking at the gut microbiota of these mice. There is a strong correlation between the body weight of wild-caught individuals and phylogenetic diversity of gut microbes, and he gets very excited about this kind of thing. So there are many phenotypes to study. I'm going to focus for the remainder on this east coast of North America because it's the transect for which we have the most data, and this is work that Megan Pfeiffer-Rixey has been doing. So these are 50 mice, 5 populations of 10 mice each. She's sequenced their exomes, and she's also sequenced their whole genomes at low coverage, and then compared phenotypes of live mice from the endpoints of the transect. And those phenotypes, some of this work was done by Katie Ferris and Dana Lynn and Mike Sheehan. So the first thing to say is that, as I mentioned before, over a longer transect, mice are larger in New York compared to Florida. So all the phenotypes I'm going to be showing you are just between New York and Florida. So very, relatively close geographically when you think about the whole North and South America. So we see differences between sexes and differences in wild mice, F1s and F2s. So we know that the differences are genetically based. We know that they're not just maternal effects. So you go to F2 or later generations and the differences persist. These mice are heavier in New York compared to Florida, and they're fatter. So BMI is different. And so this makes sense. You might expect mice in a cold climate to have a higher fat content. If you start to look at aspects of blood chemistry, you see many differences, and almost everything we've looked at shows differences between the populations. There are higher adiponectin and lower leptin levels in New York compared to Florida. Adiponectin is a peptide hormone secreted by fat tissue that affects insulin and glucose sensitivity. So human populations also vary in adiponectin levels, and there are higher titers in populations from colder climate. So there's some consistency between metabolic differences that we see in humans and in these mice. There are lower glucose levels in New York than in Florida. I'm just going through these to give you a feel for the fact that there are many phenotypic differences. And lower triglyceride levels in New York than in Florida. Here's a very different phenotype that I find interesting. New York mice build larger nests than Florida mice. So this is the phenotype that Carol Lynch studied. She was absolutely right. My skepticism was unfounded. There's an example of a nest from New York and one from Florida, and the Brazilian mice do even worse at building that nest. So this is, again, you see this in F1s and F2s. These are genetically based differences in phenotype that evolve over very short periods of time. New York mice are more active than Florida mice. So Katie Ferris built these little, she had attached a bike odometer to a wheel running machine. So this includes your two great loves, physiology and bicycle riding. And measured the activity of mice in these different populations. Okay, so what about the genetics? Let me talk first about the exome capture data. We used a nimble gene in-solution capture array to pull down all of the genes in the genome. And we have about 16x depth, so we have pretty good coverage. There are many ways to analyze these data. And when you do this sort of analysis, one always worries about population structure. So the big bugaboo of this kind of work is separating out signals of selection from underlying population structure. And there are many methods for dealing with this, and we've fiddled with many of these. At the end of the day, though, it turns out that in this case, if you just look at principal component analyses of all of the SNPs in the data, you don't see any isolation by distance. So the populations hold together. You can assign a mouse easily to an individual population, but it's not the case that the nearby populations are genetically closer to one another or that there's any isolation by distance. What it's like more is as if someone took a bag of marbles from the ancestral population in Western Europe, shook it out, and kind of spread it out across North America. So another way of saying this is you don't see clinal patterns of variation in general across the genome. Against that backdrop of very little population structure, there are genes that show very strong patterns of clinal variation, mirroring the phenotypic clines that we see. So one simple approach is to just look for those outlier genes that show strong clines, and you can do this in a couple of ways. You can just calculate correlation coefficients between allele frequency and latitude and then take the sort of tail of that distribution, or to look at the slope of those correlations and take the tail of that distribution. And in the extreme, if you take both of those, then these are genes that look very different from the background pattern of genetic structure. And so we've done that and identified, or I should say Megan has done this, and identified a whole bunch of genes that we think are outliers. And when you do that and then look at these genes, the first thing that stands out is there are almost no non-synonymous changes at those genes. And we know this with certainty because we have sequence of the entire exome. So we can simply look at the data and say there are no non-synonymous changes. Well, there's a small fraction that show non-synonymous clinal patterns, but the vast majority don't show that. And remember, this is an exome capture array, so we're not looking at intergenic regions. There's a little bit of intergenic, there's a little bit of intron, but we're mostly looking at coding regions, and yet all of the differences that we see seem to be at sites other than non-synonymous sites. And so what this tells me is that these differences are very likely to be regulatory, and these are ones that we think underlie adaptive differences between the populations. You can break this down into UTRs and introns and intergenic regions and see that you see some of these in all of them. One interesting aspect of the data is this. If you polarize these SNPs using an outgroup and ask how the clines look, so where is the high frequency derived allele, you see that about half the SNPs go in one direction and half the SNPs go in the other direction. And one interpretation of this is that there's been adaptation not just to cold or not just to hot, but to both. And if you think about Western Europe and think about the east coast of North America, so we imagine that the ancestral population is like France or England. The climate of France or England is much more like Virginia or North Carolina or Delaware than it is like upstate New York or Florida. It's not subtropical, and it's not really cold in the winter. It doesn't snow that much in England. It certainly isn't like upstate New York. And so it may be that the ancestral population was somewhere in between, and that there's been selection in both directions. Okay, well I'm getting close to the end here. Here's an example of the kind of genes that stand out. So this is a melanocortin-3 receptor, at which we see no non-synonymous sites, but we see many SNPs that show this strong correlation with latitude. If you then look immediately upstream and downstream of melanocortin-3 receptor, so this is just looking at the correlation coefficient. This is what I just showed you in a different form in the previous slide. This is the gene upstream and this is the gene downstream. What we see is the signal is really restricted to this gene. So in this genome scan, we can pinpoint a gene that we think may have responded to selection. We can pinpoint it very precisely. I'm not going to really show you the nuts and bolts of the whole genome sequencing that Megan has done, because it's complicated, but I'll just say that it's in very good agreement with what I've showed you based on exome sequencing. So if you just compare the SNPs that were surveyed in both, there's very good agreement. And if you just ask this question about how many of them are non-synonymous, well, in the whole genome data, almost everything that is an outlier is intergenic or intronic or nearby a gene, but not non-synonymous. So again, strong evidence, we think, that a lot of these differences are regulatory. So what are the next steps? Well, there are a whole bunch of obvious things. One is just to look at expression differences between these populations, and we're doing that now. So Megan has the data in hand, and I asked her yesterday if she had something I could show today, because we expect that there are going to be expression differences, and she said, well, you're not going to like this. And I said, whoa, and she said, well, there aren't as many expression differences as we thought. So the jury's out on that. But there are some, and I think especially those genes that show expression differences and also show these kinds of clinal patterns of variation at genes that are reasonable candidates for the phenotypes that we've identified as being different are ones that are good to look at. So that's one direction, and then the other direction is to actually do QTL mapping. So Katie Ferris has set up some crosses between, not Florida and New York, but between Brazil and New York, which are even more different, and is making a large F2 mapping population. And the goal here is to try to map genes that underlie many of these phenotypic differences, and then overlay those QTL map with results like this, where we have individual genes that really stand out. And the hope is, by doing those two things together, we can pinpoint individual genes that we think have responded to selection over short evolutionary timescales. Okay, so for the second half, house mice in the Americas show clinal patterns of variation for many traits. Consistent patterns of covariation with the environment in different transects suggest that these traits have been selected. I didn't actually talk about this, but part of the aim behind this whole project is that if you look in both North America and South America, and you see similar clines in opposite directions as we do for body size, that's good evidence that these are under selection. And so that's been part of the motivation. Trait differences among populations persist in the lab and therefore have a genetic basis. Most genes don't show clinal patterns of variation, but some do, and that really suggests that those have been subject to selection. Most adaptive evolution in these populations seems to be due to changes in gene regulation, and we have some strong candidates that have been identified. And the nice thing about these are mice, and so they're really amenable to functional tests of all sorts in the lab. I just got an email today from Russell Vance at MCB saying they've got CRISPR working in mice to not only knock out genes, but to introduce point mutations. So that's the kind of thing we hope to do. So I just want to return to our good friend, the chimpanzee, and our friend Donald Trump, and remind you that evolution consists of these two aspects, the splitting of lineages and adaptive changes along lineages. King and Wilson suggested that these kinds of adaptive changes are probably caused by changes in gene regulation, And I think that there's good evidence for that now, but I think there's also good evidence that gene regulatory changes are important in speciation. I think I've mentioned everybody as I've gone along, but these are the current lab members and some of the people in the lab who previously worked on mice. Michael Sheehan, Jeff Good, Brett Pacer were some of the ones that really contributed. So I'll stop there and take questions. Thank you. And maybe we'll let the people who need to get out sneak out first. Caroline.
So,
phenotypic.
Go ahead.
okay. I've kind of got two, but I can't decide which one. Okay, so the pattern where the derived allele is at 50%, there is high frequency at either end of the climb.
Yeah.
Is that also potentially consistent, not with adaptation to heat, but with relaxation of selection related to cold hardiness? Because you'd expect alleles that promote fecundity and reproductive success would rise just through a relaxation of requirement for winter ability.
Yeah, I mean, if there's a cost, so I mean, that's a trade-off, and so in that case, the selection might be on some reproductive trait which exists in a new environment when there's not some sort of constraint on it otherwise. I didn't say this, but there's a huge seasonality to mouse breeding in the high latitudes and extreme southern latitudes, but not in the tropics. So if you go to Brazil or even Florida, you catch reproductive females at all times of year. That's certainly not true in New York. Now, how much of that is phenotypic plasticity and how much of that is genetic, we don't know. We can breed them year-round in the lab. But there is this interesting observation that many mouse breeders have made, which is that even in the lab where you're controlling day length and temperature and sometimes humidity and everything, there's sort of a seasonality to breeding. So the mice are somehow detecting the seasons. It's kind of a mystery how that's happening. Paul?
So, if you don't know exactly how many times mice were brought to the United States, I wonder, how do you tease apart local adaptation from just the hypothesis that they, coming from different genetic backgrounds from different introductions across Western Europe? And I was wondering, could you make that same map, including all your, that PCA thing, including the European sources as well, to see if maybe there is isolation by distance? It just corresponds more to the ancestral lines than...
Yeah, no, it's a great question, and something I'm very interested in. So we're collaborating with Jeremy Searle to survey populations in Europe, so we can look at this in the context of Europe. So the extreme version of that would be, what if mice from North Africa colonized Florida and mice from far north in Britain colonized New York, and they were adapted to those differences? It's not selection in situ, they're genetic differences, and they've just come together. The evidence against that, well, if that were the case, you would expect to see some kind of isolation by distance. You'd see something that looks kind of like secondary contact, and we don't see that at all. So we don't think that's true. The other thing is that in Europe, the standing levels of genetic variation are very similar to what we see in North America, and even the alleles and haplotypes that we see are similar. So my guess is that that's not what's happened, but a long-term, so there are two important long-term interests in this regard. One is just to sort out the demographic history of the colonization of all of the Americas, and you can imagine all kinds of simple, kind of fun hypotheses, like the mice from Brazil are most closely related to the mice from Portugal. The mice from Argentina are most closely related to the mice from Italy and Spain, because Argentina is kind of half and half. The mice from Quebec are like French mice, and the mice from the United States are like mice from Great Britain. So, and you see that kind of thing with other histories of colonization around the world. So that sort of tying it to the ancestral populations is one, but then the other is just actually looking at doing some demographic modeling to try to get a sense of bottleneck sizes, and so we haven't started on that yet. There was another question. Russ?
So the Klein stuff you presented in the first half of this talk, it seems like there's a sort of asymmetry with the way selection is going to operate here, in the sense that one of these is very bad in a hypergenic background. The other, the other X chromosome segments has really no effect as far as you can tell.
Yeah.
So this suggests to me you should actually have a Klein that reflects that, where you'd see lots and lots of introgression in one direction and then just sort of truncating right at that hybrid zone. Do you see anything like that, or is that?
Yeah, so we've looked carefully for that. The X chromosome shows a little asymmetry that way, but it just doesn't introgress very far. Neither the X
Right.
nor the Y introgress very far, but there is a little asymmetry there. And then for the autosomes, you see asymmetries that go in different directions.
Mhm.
Yeah.
I'm trying to think about your X inactivation and gene expression results. And I was kind of trying to wrap my head around like, what's the mechanism for X inactivation here, because it seems like you have more and more of the X coming in, and you have more and more hybrid problems. But I would have think that it's either it's like the whole X is inactivated and not piecemeal, or it's kind of
Yeah,
I'm
so
trying to wrap my head around that, because the expression changes were like so global.
yeah, and that's true. And so I didn't go into great detail on this, but we have tried to map that. And what I can tell you is that there seem to be many genes distributed along the X that affect X inactivation. But if you take a little piece of the musculus X chromosome and you put it on a domesticus background, so let's say here's the X chromosome, and you take just this piece here, the entire X has a disruption of inactivation. So what that suggests is that there are trans changes that map to the X that affect expression along the X, and there seem to be a number of those. It's a very complicated process, and we're starting to learn more, and I'd be happy to chat about it. It gets detailed very quickly. But we don't, and the exact genes that are involved in X inactivation, only some of those have been identified.
So in these situations where you're finding these genes where there's basically this trans effect and there's this compensatory evolution, so is that's implying that there's been evolution of both the regulatory region and the expressed gene? Is that correct?
It's suggesting that there's been evolution of something that binds, so a transcription factor that binds to assist regulatory
So it's really
and
just the expression factor plus the receptor.
It's those two things that have both changed. And the cool thing is you look at the two species and you don't see any evolution. If you just looked at them, you would think there's been no change.
Yeah.
But then when you dissect it, you see that there in fact has been a change. And so the model is there's probably selection to maintain a certain level. Well, the model that I favor is the following. Imagine that there's selection to increase the expression of some gene for whatever reason. And the first mutation that comes along is one that changes a transcription factor. And that transcription factor now upregulates that, increases the expression of that gene, and that's a good thing. It's favored by selection. But transcription factors are pleiotropic. They act on lots of genes. And so, maybe there are a whole bunch of other genes that are now also upregulated, and that's all bad. But the benefit of the good thing outweighs the detriment of all of those bad things. But then there's selection, compensatory evolution, to adjust all of those things that got tweaked that are not favored. And so if that's kind of the way a regulatory evolution evolves, you could imagine that there's just a lot of compensatory evolution all the time. And then you bring things together in a hybrid and things get knocked out of whack and cause sterility. Yeah, Jim.
So is X inactivation or Z inactivation, is this like common to everything with sex chromosomes, or is this unique?
If you had asked the question 10 years ago, or 8 years ago, I would have said yes. And then David Pressgrave sort of threw a wrench in our thinking about this as far as Drosophila goes. So in Drosophila, the mechanism of X inactivation is There is no identical mechanism of X inactivation in Drosophila, but it is the case that genes that are put onto the X chromosome in Drosophila are in a sort of repressive regulatory environment. And the experiment that was first done, you know, again, it gets complicated, but if you take reporter genes and you put them onto the X chromosome in Drosophila, you see that they're expressed at a lower level in male meiosis after pachytene than the same genes that are put on autosomes. So that sort of looks like X inactivation, but the mechanism of it is different in Drosophila.
Does that mean that we know that in some lineages they don't have this, or do we just not know for the majority of species? I'm thinking about this from the standpoint of whether it affects the rates of speciation in groups that have X inactivation in place and species that don't, if there are species that don't.
Yeah, so it exists in birds, we know that. Yeah, that's an interesting question. I don't think anybody's looked at it, but I don't think we really know, but I mean, so it's been done in chickens, Drosophila, and mammals. But it's actually something you can see cytologically through the microscope. If you look at the right stage of meiosis, you can see this sex body that forms. It's all heterochromatic and becomes what's called heteropyknotic, so meaning just that it stains darkly in the microscope and is associated with, you know, basically coiled heterochromatin state that prevents transcription of those genes. So, you know, in principle, this is something that could be studied. It's interesting. Yeah.
Very interesting. Thank you. What are the candidate transcription factors and cofactors, and how do they vary with the environment?
Yeah, so we don't, in the experiment that we've done, where you make an F1, and you can say that there's been compensatory evolution involving both a cis change and a trans change. At the end of that experiment, you know where the cis regulatory element is. It's at the gene that you've surveyed. You don't know where the transcription factor is. And so the only way you can get at that is to actually do a mapping study in like an F2 mapping population. And even there, it's you don't get very close to the gene, so we don't know. Michael.
I have two questions. One, do you see any compatibility between the different populations along the Clyde, like Florida versus the New York? Do you see any incompatibility?
Oh, we don't know. Yeah, that we haven't done those crosses yet, and that's an interesting question.
And taking that back to the first project,
Katya wants to do that experiment, by the way.
Yeah, because I read this study where they were looking at the hybrid zone, looking at mice, and looking at their microbiotas, and suggesting that one feature of the incompatibility between different species might be adaptation of their microbiota, and then that creates the incompatibility. So I was wondering if you see any indication about things like that.
Yeah, we haven't looked yet. So Katya and Taichi will answer you in a they'll figure it out. All right, well, maybe we should stop there. If people have more questions, feel free to come up and chat with me. Thanks.